So, good afternoon. I'm really pleased to introduce today's speaker, Isa-Rita Russo. Isa-Rita is a senior lecturer at Cardiff University whose research focuses on evolutionary genetics and conservation. She investigates how landscape and environmental factors shape genetic diversity and population structure in wildlife using genetic data to inform conservation and management. Her work encompasses a range of species, including rhinoceros, leopards, and servals and hartebeest, and applies molecular tools for microsatellites and SNPs to whole genome sequencing, alongside phylogenetic, population genetic, and bioinformatic approaches. So without any further delay, I'll hand over to today's speaker, Isa-Rita. Thank you very much, and over to yourself.
Thank you very much. Do you hear a lot of background noise because they are drilling outside my office and it sounds horrendous in my office, but do you hear background noise on the microphone?
We did before, but it seems to be quieting down when you start talking. So long as you keep talking, we'll be fine.
Okay, perfect. Okay, so thank you very much for the invitation. Maybe I can just start with how I think I was invited to do this science talk. I'm at the moment working with Teresa Robinson. Most of you know her as Tree, and she is someone that's involved in the indicator work, which is one of the indicators, genetic indicators that the CBD wants countries to report on. So I have been helping the UK government basically to generate genetic indicators for a number of species. And for this, we went to Scotland. And we did like sort of a hackathon of 3 days, just sitting in a room and trying to go through a list of species and then, find the relevant information for these species and then calculate the genetic indicators for those species. And then all the data went back to JNCC, and then they did sort of the final statistical analysis on that data set. And that's really how I met someone working for JNCC, and then a few months ago, then she invited me and she asked me, would I like to give a talk? So that's where sort of my connection with JNCC is from. I'm going to talk about something very, very different today. As you can see from my title slide, I'm going to talk about African rhinoceros. I'm going to ask you maybe if you can switch on your microphone if you know the answer to this question. And I have a question for you. Do you know what rhino we have on the screen? Because we have 5 species of rhinoceros. I'm just going to talk about the African rhinos. In Africa, we have black and we have white rhinoceros. So can you tell me which of the African rhinos do we have here on the screen in the picture? Is there anybody in the audience that knows the answer to the question? There's a hand. Yes. Can you please unmute yourself and tell me, and then also explain to me why you say what you're saying?
I believe that's all. Katie, you're very quiet.
Sorry, I'd put the mic up, that's why. I think this is a white rhino, and I think it's because the name white rhino comes from something odd and a mistranslation of a wide lip. And it's got the wide mouth.
Ah, you are spot on. I thought I was going to teach you something today, but no, you are teaching the whole audience. Yes, that's exactly right. So the name white was actually mistranslated from white. As you can see here, can you see my laser? You can see here at the bottom, the lip is extremely wide lip, and that's where it came from. But that got mistranslated into white. And then if the one is in the white rhino, then of course the other species should be a black rhino. But no, black rhinos are not black and white rhinos are not white. It's just this mistranslation. And black rhinos, then opposite to what you see here in this picture, they have a very, very pointy lip, because white rhinos are actually grazers and black rhinos are browsers. So they eat leaves, so they use this pointy little lip to, to pick the leaves off the branches. So I'm really going to focus now for the rest of this talk. I'm going to really just talk about black rhinos. And I'm going to try and explain or show to you guys how we can use genetic information, which is more or less the traditional stuff that we started off using in conservation genetics, and how we can use that in sort of in comparison to genomic information, and how the two can actually inform each other and actually give you maybe at the genomic level even finer scale resolution. But generally, you would expect to see sort of very similar answers. A lot of people say we should not use any traditional genetic methods anymore. We should now just move on to whole genomes. But because I am from South Africa originally, I understand the challenges that developing countries have, and it's not always possible to do whole genome sequencing because it is still quite expensive in developing countries. So there's still a place for these more genetic, the more traditional genetic markers to be used in the conservation of animals. And we can use a combination between these sort of data sets, and we can, answer a variety of research questions. It could be either at sort of a phylogenetic or phylogeographic scale, or you can even fine-tune it all the way down to the population level. So in this presentation, as I've already said, I'm going to use black rhinos to try and illustrate this to you. And then I will start the talk off with a bit of background, just background on black rhinos, and then I will talk a little bit about the genetic markers that we've used in this study, and then from there on I will then move on to the results. And this is work that I've been doing as part of the Conservation Genetic Specialist Group. I am the coordinator for the African node within this Conservation Specialist Group, Genetic Specialist Group, and within that node, the African group calls themselves Congen Africa. So I will show you right at the end, it's not just me that's involved in all of this work. I have collaborators at other universities and other institutions as well. And of course, I'm based at Cardiff University, and I'm a senior lecturer here. So maybe just a little bit about black rhinos. And well, actually, this is rhinos, both black and white rhinos, and this is specifically data for South Africa. This is data that I actually pulled down from the WWF website, but you can see here, this is been very well documented, the poaching of rhinos, especially sort of in early 2010s on to basically 2020, that 10-year period. You can see here that the numbers of poaching went here from 1,004 individuals in 2013 all the way up to 1,215 individuals in 2014, and then from there on it came down, and the last recorded data we have, of course they probably have more data now, but the last time I have actually looked at this was in 2022. And there was still 448 rhinos just in South Africa being poached in that year. So this has been very, very well documented. Apart from this, also, the western subspecies of black rhino was declared extinct by the IUCN, and this subspecies was declared extinct in 2011. So yeah, black rhinos have had a really, really tough time. Now on to something a little bit different, and now I'm going to just a little bit talk about taxonomy within rhinos, because this is one of the things that we are still trying to figure out. So how can we actually conserve black rhinos if we really don't understand the diversity within the species? And so this diversity is below the species level. So this could be subspecies, it could be evolutionarily significant units, it could be management units, you can call it whatever you want to call it, ecotypes, but we need to understand the diversity that we have within a species in order for us as conservationists to preserve or to conserve all of that diversity. So way back in 1967, this man here, you can see there's a picture here, Colin Groves. He was an Australian and British biologist. He actually used, you can see he's actually got a skull here in his hand. He actually used skull morphology and also a cranial morphology. And he described, as you can see in the slide, 7 subspecies of black rhinoceros across the African continent. So you can see here a subspecies in the south called bicornus. We have minor, that's got a really wide distribution in this sort of yellowy color, Joviensis, Longipes, this is the western subspecies that I've just pointed out on the previous slide, that's been declared extinct by the IUCN, and so on and so forth. But in 1987, this is the African Rhino Specialist Group, they've actually adopted a 4 subspecies or 4 ecotype classification. And these are the people really that actually were in charge of that African Rhino Specialist Group. This is Roald Dway here at the bottom. Then you have Richard Emsley and Michael Knight. And they were sort of the chairs and the co-chairs of the African Rhino Specialist Group at the time. So they basically said, no, we do not agree with the 7 subspecies, but we are going to go with this 4 subspecies classification. And this basically will allow us then to sort of group these 7 subspecies into 4 subspecies. So we have Pecornus here, we have Minor here, we have Macaulay, which is an East African subspecies, and then we have Longipes. And ever since 1987, all management, all conservation of black rhinoceros across the African continent has been based on this 4 subspecies classification or 4 ecotype classification. So you can see here the Longipes subspecies is sort of the West African subspecies. That's the one that's gone extinct. Then you have this Pecornus, Diosaurus pecornus. This is a subspecies that you have in Namibia and sort of the western part of South Africa. So this has generally been regarded as sort of a dry adapted subspecies. Then you have the widespread minor subspecies in this like little dotty background, and they go all the way from South Africa all the way up to Kenya, Tanzania border. And then you have the East African subspecies, which is mainly based in Kenya, but a little bit, you can see some of it comes into Tanzania and then also in Rwanda. So those are the 4 subspecies. And as I said, all conservation has been based on these 4 subspecies. So a lot of movement has happened with regards to translocations, black rhinoceros translocations. They've moved a lot of rhinos around in Kenya. And you will see later on why this is really important. But they've also moved individuals all the way from Zululand, which is in KwaZulu-Natal, one of the provinces on the east coast of South Africa, to the Luangwa Valley, to the Shire Valley. They've even moved individuals to the Kagera Valley, which is basically here sort of Lake Victoria, because that Kagera river runs into Lake Victoria. So that's where Uganda, Tanzania, and Kenya basically meet each other. And then they've also moved individuals from the Kaoka land, as you can see here, that is in Namibia, the northern part of Namibia, all the way down to Addo Elephant National Park, which is here on the east coast of South Africa. So they've moved a lot of rhinos across these subspecies boundaries. And this has been what has been happening ever since, you know, rhino conservation is basically started in Africa. So this has really been done without really an understanding of really what is the actual underlying diversity. So it was 7 subspecies and then it was just amalgamated to 4 subspecies, but no one has ever really studied this. So that's sort of the rhino background. Now I want to move on to the DNA, because I'm a geneticist, so I use genetics, genetic information to inform the conservation of mammals in general. So I just wanted to point out, maybe this is to some of you will be, you know, common knowledge, but let's take all of us back to A-level biology. We have 2 genomes. So this is a cell, as you can see in this picture. We have these organelles in our cells, these little orange organelles, they are called mitochondria. And these organelles have their own genomes. They are circular genomes, and they are about, you know, in humans, mammals, about 16,000 base pairs in length. So we can actually use the mitochondrial genome, but then we also have another genome, and that is the genome that's sort of situated here in the nucleus of your cell, okay? And these are the autosomal chromosomes and also the sex chromosomes, okay? So this DNA information is carried on chromosomes, It's carried by the chromosomes. So you have autosomal chromosomes, and then you have sex chromosomes, okay, if you're like us mammals. And then you have the mitochondrial genome, which is a circular molecule. It's a circular genome, double-stranded genome that's situated in the mitochondria. And we can use both of these different genomes to inform the conservation of animals. Remember that the building blocks of DNA is A, T, C, and G. So you will see here is a DNA molecule. So it's a double-stranded molecule, and you have A, T, C's, and G's. So it's made up of A, T, C, and G's across both these genomes. And I will talk about the A, T, C, and G's in a minute, just to describe some of the markers that we've used. But in this study, we've actually used mitochondrial DNA, and we've also used nuclear DNA. And at the nuclear level, we've used basically just gene regions, but we've also used whole genome information. So initially, when we started this project, it was many, many years ago when I just arrived actually in Cardiff. We really started with sort of the traditional sort of genetic analyses. In those days, we used mitochondrial DNA from the mitochondria. So those were just like gene regions that we would analyze. So 400 or 500 base pairs. And then we would use those in phylogenetic trees. So we would like draw little trees to see what is the relatedness between the different individuals in our populations. And we would then say, yeah, these individuals are all clustering together. Oh, so they are all south of the river, and these individuals all clustered together, and they are individuals north of the river. So you can start seeing the patterns then emerging. But then we also then in this study, we use what they call microsatellite markers. So this is the thing that people initially started using when they started looking at population genetics, is microsatellite markers. So these markers are just tandem repeats. So you remember I said DNA is made out of A, T, C, and G. Those are the alphabet letters for DNA. So they repeat themselves. So you can have a repeat, for example, of 2 base pairs. That's a 80, 80, 80, 80 repeat. Or you can have a 3 base pair repeat, ATC, ATC, ATC. And the difference between me and you, or one rhino and another rhino, is that the number of repeats between ourselves would be different. So I will have 10 repeats, 80, 80. You will have 15 repeats. The next person will have 20 repeats. And based on those number of repeats, we can actually then go all the way down to the individual level. So we used a higher level mitochondrial DNA, which was just long base, long sequences for 500 base pairs. But then we also used the finer scale microsatellite markers, which is basically allowing us to drill down to the individual level. And that's exactly what they use today in forensic analysis. If you've watched CSI, they use a set of microsatellite markers. So you don't just use one microsat, you use 20 or 30 microsats that's scattered across your genome. And building those profiles then together, you can then actually say, this is individual 1, this is individual 2, because each of those microsatellites will give different repeats depending on the individual. And that's why you can then basically drill down to the individual level. So in this study, we really wanted to understand the extinction that we've seen in black rhinos, because we know black rhinos has been hit hard by poaching. And also we wanted to look at the genetic erosion that has happened then subsequently, and what were the conservation options then for black rhinos. And up until this point, all the previous studies were geographically restricted. They also had very restricted samples, and then they assumed this 4 subspecies classification that was adopted by the African Rhino Specialist Group. So it was really important way back, this was like sort of the early 2010, 11, around there, to understand the range-wide genetic diversity that existed also prior to the population collapse, because once you understand what you have lost already, it could actually inform you how you should go then forward to conserve what is left, okay? And is there actually opportunity to maybe get back some of that genetic diversity that we've already lost, okay? So understanding historical diversity will then, of course, assist you in conserving the extant diversity. So in this initial project, we actually collected a lot of museum samples. So museum samples basically only exist in museums now. They don't exist in the wild anymore. And so we could actually then paint a really nice picture, historical diversity versus the extant diversity, which is what we have right now still in the field, and also those museum samples then put together. And we wanted to compare those two diversities to understand what have we lost and how can we go then forward with the species. So then I'm going to show you the first result. This is the mitochondrial DNA result. So as I said, we have 400 or 500 base pairs of a specific gene region from the mitochondrial genome. So you can see here, here's again a cell. We have these little organelles. They are mitochondria. We call them the powerhouse of the, of the body, of the organism, because they generate all the energy that we need. And you can see here we have these mitochondrial genomes. They are circular molecules and double-stranded. And from this molecule, we can then sequence specific gene regions. And the first thing we generally do is we do a phylogenetic tree to look at the relationship between individuals that we have collected and see if we can actually see patterns emerging. So the first thing I want to show you is that all the lines in red have gone extinct. So you can see here we have 64 lineages, okay? These are all little lineages, okay? Unique variation that we have within black rhinoceros, but 44 of those has gone extinct out of the 64. Then if you want to just look at the overall patterns, you can see here we have a Southern Africa group. Can you see it here? Within Southern Africa, we have then finer scale groupings. We have the Southwestern, the Southeastern, and then the Southwestern, again, it's a bit scattered, that one. And then we also have the South-North group. And then we have what we call an East Africa, Central African group. And that includes 3 smaller groupings, the Central Africa, East Africa, and the Chari Victoria group. And then we also have what we call the Northwestern, Northeast African group. And then right here at the bottom, we have the Western group. And that is the subspecies that's been declared extinct. You can see both these two lineages or variation that we have within that grouping. They are both gone extinct. So we don't have that subspecies anymore. It's gone extinct. This is what we call a haplotype network, and it basically shows a very, very similar picture. You again have your East Central Africa group, you have a Southern Africa group, and then you have this Northwest, East Africa group, and then right here at the bottom, you have the actual extinct Western subspecies. So that was sort of a very initial result. Then from there on, we moved on to the microsatellite markers, these tandem repeats, and now you can drill down more to very individual, individual level. So each of these lines that you see, for example, here's a line here, is actually one individual, and you can see sort of the makeup of that individual's genome. So you can see here that this individual is a bit red, there's a bit of blue and a bit of orange in this individual. So these different colors basically shows us different genetic backgrounds. So remember, now we are talking here about sort of the microsatellites that are situated within the nucleus, which is the DNA that we have that is basically compacted into the chromosomes. So again, we see a very, very similar result here compared to the mitochondrial DNA, where you have like a Southern Africa group, and then you have the Central, East, North, Eastern, and Northwestern Africa group. So it seems here that Southern Africa always splits away from the rest, okay? And that's potentially to do with sort of the geography and the rivers that we have. So it seems that the Zambezi, the Chobe River seems to be a barrier to gene flow. And this is not just for rhinos, this applies to a lot of other mammals and other animals across the African continent. Zambezi, Chobe River seems to be a barrier. So south of the Zambezi is always a grouping, and then north of the Zambezi is another grouping. So in south then we have the individuals which are from South Africa. In this case, they were all from KwaZulu-Natal, specific province in South Africa. We also have the Namibian individuals, so the Khoekhoeland individuals, and then we also have the individuals from the Zambezi and from Zimbabwe. And then on the other side, north of that river, we then have individuals from Central Africa, which is Eastern Africa, which is basically all the Kenya individuals, the Masai Mara individuals, and then we have the northeastern and the northwestern individuals here, as you can see here, Chad, Cameroon, and so on, and then Uganda and, South Sudan. These are just two different analyses. So if you use one analysis here, it's not as sensitive to the data. But if you use this, this is called structure analysis. If you use this one, which is called a BUPS analysis, you can actually see that another grouping pops out here in the yellow, which is the Chari Victoria grouping, but you don't see it when you use the structure analysis. So basically there's 5 to 6 genetic groupings within black rhinoceros. Okay, this is a combination between the historical samples and the modern samples. And we did this on 11 microsatellite markers, and it was a data set of 560 individuals, the biggest data set that's ever been used across the globe, or across the African continent. And then the other thing I wanted to look at is actually the how did the genetic diversity then decline? And we can use something called expected heterozygosity here. And in general, the expected heterozygosity decline. One of the groupings, if you look at the South Central grouping, expected heterozygosity went from 0.74 to 0.53. So just to show you quickly a sort of a table showing some of that result to you. This is the historical data set, and this is the extant data set, which also includes the historical samples. But you can see here in a country, just Kenya, just looking at black rhinos in Kenya, this heterozygosity has actually gone from 0.74 all the way down to 0.7. If you look at Tanzania, it's gone down 0.67 to 0.64. But then if you look at bigger groupings, not just countries, you look at South Central, okay, then you can actually see that it's gone all the way from 0.74 to 0.53. So this is actually the subspecies minor, okay? So these are the 4 subspecies based on the regions. So South Central is the minor subspecies, the one that goes all the way from South Africa, the widespread subspecies, all the way up into Africa to East Africa to the border of Kenya and Tanzania. So you can see here that we've lost a lot of heterozygosity comparing historical samples with the extant samples. So we've definitely lost a lot of genetic diversity in the species. And we all know that if you lose genetic diversity, it will potentially be harder for that species to adapt to climate change. So I always say to my students, genetic diversity equals options. You have options. If you have genetic diversity, you have options to adapt. You can either go this way or this way. If you don't have genetic diversity, you kind of forced into that bottleneck, and there's only one way, and it's going basically downhill. So that was sort of the more traditional genetic analysis that we've conducted for these rhinos. And then, of course, the era became the era of the whole genome data analysis arrived, and of course now we had whole genomes available, and it became cheaper and cheaper and cheaper to analyze whole genomes. So we really wanted to use the whole genome data set and to see whether we had the same answers with the whole genome data set, or could the whole genome data set actually find some of sort of the questions we still had with regards to the diversity that we see in black rhinos across the continent. So we wanted to really expand on our previous work by taking advantage of genomic sequences, and we wanted to try and see if we could resolve some of the patterns that we could not resolve with just the traditional markers. And we also wanted to look again at population genetic structure, and we wanted to look at gene flow across the African continent to see how populations were connected and still are connected to this day. And we wanted to also evaluate whether the modern samples that we still have are representative of the historical diversity from same geographical regions. So in this study then, I think we had a total of 83 or so samples, I think in total. 63 samples, and of the 63 samples, we had about 20 samples that were modern samples. So you can see here in this little graph, we had historical samples, those are the samples in the purple, and then we also have modern samples, and those are the samples in this sort of pinkish color. And you can also see that the size of these circles, actually they indicate then how many individuals we've collected. So most of our modern samples were based sort of here in East Africa, but we also had some modern samples. You can see here is one from Namibia, and then we also had a sample here from KwaZulu-Natal in South Africa. So we had sort of a representation of a mix between the modern and the historical samples. And here we again basically only used the nuclear DNA, so the DNA on the chromosomes, but now we've basically sequenced the whole genome. So 2.5 billion base pairs in length, okay? And from this whole genome then, you then have an opportunity to actually go and pull out where you have differences between individuals. So here is just an example to show you at the bottom. So you have 3 humans here, purple, the blue, and the orange human, and you can see the DNA, and you can see at this specific position, you can see in the purple person, we have an A, in the blue person, we have a G, and in the orange person, we have a T. And this is basically then what we call a SNP, okay? A SNP is a single nucleotide polymorphism, okay? So that means that we have just one base pair, and across all your individual, we have differences in that specific position. So you can see here, all the rest is the same. It's a C, a C, and a C. And this is what we generally do when we actually look at whole genome sequencing. For whole genome sequencing, we generally basically look at these differences, these single nucleotide polymorphisms across the genome. And that you then pull out, and then you base all of your analyses on those differences between different individuals. But this is the whole genome. So you sequence 2.5 billion base pairs, and you end up with, I don't know, 2 or 20 million SNPs, 2 to 20 million SNPs, and then you clean that data, and then you end up still with, you know, millions or maybe thousands of SNPs in the end. And one of the first things that people do is they do this analysis called a PCA. This is a principal component analysis. And for this analysis, then you can see here, we have a principal component 1, a principal component 2. And the first thing that is really striking, if you look at this, is you can see here that this is the southern samples, so south of the Zambezi and the Chobe River. So they actually pull out, if you look at PC1, from the rest of the samples. And then if you look at PC2, you will see these samples here from Chad and Cameroon, so sort of, you know, West Africa and Northwest Africa, then they pull out from the rest of the samples. So because we have this really then strong split between Southern Africa and the rest of Africa, you know, this Zambezi Chobe sort of, can I say, barrier or divide, we then decided to actually analyze all of the data that you can see here and split it by north of the Zambezi and south of the Zambezi. So basically what I want to show you here, I'm not going to go into all the details, but I just want to show you a few things here. We have historical samples, as you can see here with the little circle. So here's some historical samples, and then we also have modern samples, and you can see here all the modern samples here are indicated by the little triangle. So we only had 20 modern samples to put this in the context of the modern samples. Most of the samples that we included in this data set were historical samples from museums, so museum skins generally. And you can see here what we actually then can see is different populations. So you have the Northwestern, the Northeastern, East Africa, Central Africa, and here is the modern samples, because this is a mix between East Africa and Central Africa. And then you have another population here in this light pink, which is the Ruvuma population. So this population also pulls out quite quickly, and I will show you now where this population lies in relation to all the other populations. And then again, this Northwest population, which is the Chad Cameroon individuals, they then pull out at PC2. So a very similar pattern. And then when you basically just zoom into the south and you just look at the south, you can see here the first thing that pulls out here in the south is these Angolan samples. So these are the samples that are sort of towards the west in Namibia. So the western part of Southern Africa will pull out first, and that is generally the arid adapted rhino. So they pull out from the rest. And then later on, some of the other groupings will pull out. But there's definitely, as you can see, within the species as a whole, there's a lot of, finer scale diversity. And it's really important to conserve all of these diversities as a whole, because this will allow rhinos to be able to adapt in the future. Then maybe just quickly a phylogenetic tree again. So you can use the DNA on the chromosomes, and you can actually divide it into 10,000 base pairs, and you can then use blocks of that across the genome, and you can do these trees. But basically all I want to show you here is that this Rovuma population, which is in the southern part of Tanzania, northern part of Mozambique, this population is actually closer to the southern groupings, which is south of the Zambezi, than it is actually to the rest of the African population. So there's a close relationship between the southern, populations or the southern groupings and this Rovuma population. This is actually a population that we still don't know if we actually have rhinos in the southern part of Tanzania. No one has seen rhinos for many, many years, or the northern part of Mozambique. So this is actually an area that we would like to actually go back to now and do some proper surveys to see if there are actually still rhinos, because you can see there's a very, very unique diversity in that part of Africa. And then if we do a sort of a similar analysis as I've shown you with the traditional microsatellite markers, you can see here when you compare, you see again, you have a southern group, this Rovuma group, which is in the southern part of Tanzania, northern part of Mozambique, Central Africa group, East Africa group, and then you have the Northwest and the Northeastern group. And then you can actually ask the program to give you finer scale structuring if you want. So you can actually push it up to how many Ks, which is the number of genetic clusters you want to test. But in this case, we tested up to 10 clusters, and you can then see if you force the program to give you 10 clusters, what happens in the southern part of Africa. You have the southwestern grouping, which is those desert adapted rhinos. They will pull out. Then in Central Africa, you will have Maasai land, which is very unique rhinos. Historically, they are very different from all the other rhinos in Central Africa. And you also have Eastern Rift rhinos in this light pink. And then in East Africa, you have Northern Rift grouping. And then you have, again, your Northeast and your Northwest. So you have 6 populations, and then you can actually push it down to a further 4 subpopulations. So in total, if you want to really look at all the variation and the fine scale variation within the rhinos, you can go all the way down to 10 sort of subpopulations. Okay, and then we just wanted to quickly look at migration. How does migration happen? So this is basically a program that we use to test for migration across sort of a landscape. And you can see here where we have sort of the gray colors. This basically shows that these areas are sort of barriers to gene flow. And then where you have the blue colors, that's where you have higher, than average sort of migration, than expected migration. And you can see that these sort of gray colors, basically, they correspond quite well with the Albertine Rift, the Ethiopian Rift, or the Gregory Rift. So it seems like, all these natural features, they've sort of helped to shape the diversity as we see it today, okay? And some of these rivers have also been important in shaping the diversity of rhinos. So again, you can see that big sort of barrier here. Here's the Zambezi Chobe River. You can see there's a big barrier here. So everything to the south is quite different from what we see to the north. So there's been some restricted gene flow or no gene flow for thousands and thousands of years, okay, between sort of the southern populations and then the populations further to the north. And then with each of these kind of bigger groupings, we have then finer scale structure. So then if you just compare, the original data sets to, the whole genome data set, it basically shows you that there's very, very good correspondence at a higher level with the 5 to 6 sort of, initial groupings. And then you can if you want to go finer scale, then the genome analysis, whole genome analysis allows you then to go finer scale, and you can actually then pull out, those further 4 subpopulations. But we can't actually pick these further 4 subpopulations up when we actually just use the traditional microsatellite data sets. So I would say they give you more or less the same answer, whether you use traditional genetic markers or you use whole genome data, but the whole genome data can actually allow you to zoom into very, very fine scale structure, and also it helps us really in this case to define some of the boundaries between some of these different genetic clusters. There is a reference here to a paper, Bertola et al. 2024, and this is a paper that we've published a few years ago, basically talking about, these different genetic data sets and different genetic markers that people are using. And in this paper, we actually use black and white rhinos as an example, where we had data sets, traditional population genetic data sets versus whole genome data sets, and basically just illustrating that they actually give you very, very similar answers. But in terms of the whole genome data sets, it actually sometimes gives you further finer scale resolution. And there's some other individuals or some other species in this paper that we also refer to, that if you're interested, you can go and read up on those. But basically just showing that there is still a place and a time and space for traditional sort of genetic markers. In conclusion then, I think, by using the actual whole genome data, we had support for these different groupings, the Northwest, the Northeast, the Rovuma, the Maasai, East Africa, and the Eastern Rift and the Northern Rift, which we haven't had when we actually used the traditional. So this is more the kind of finer scale resolution that we were allowed to actually observe using the whole genome data. And this actually whole genome data also allowed us to be more precise in terms of, the localization of populations. For example, we know now that the East African sort of genetic cluster is between the Albertine and the Gregory Rifts. This Rovuma population, which is the population that we have in the northern part of Mozambique, southern part of Tanzania, is a very, very important population. And it places enormous conservation value on any of its remaining individuals, because, as I said, they haven't seen rhinos there for a very, very long time, but there's a very, very unique genetic diversity that's been, been captured within that population. So it would be good to go back to that part of Africa and do more surveys. So basically this will basically include the Selous Nature Reserve, and then also the Niassa Reserve in the northern part of Mozambique. Then I didn't talk too much about this, but basically I have shown you that the Maasai, Mara, Serengeti populations are quite different from the rest of the East African populations, and they should be managed separately from the rest of Kenya, because Kenya now is basically a meta population where they've actually mixed East African and Central African individuals. I've showed you in that initial slide the mixing that's gone on in East Africa, in Kenya specifically. And so they've basically mixed two of these genetic clusters with each other. So it's not really, pure genetic clusters anymore. It's now a big mix. Kenya is kind of a meta population, or a mix now between this East African and Central African grouping, but the Maasai Mara is quite different, and that should be definitely managed separately. So the genetic and the genomic data basically complement each other, and the genomic data gave us improved resolution. So the next step for us will be to go back to Rovuma. So we want to do some surveys in that part of Africa to go and see if we actually do still have rhinos in that part of the continent. But also the next focus will be Kenya, because Kenya is definitely the stronghold of genetic diversity. A lot of diversity comes together. You have the Maasai, you have the East African, you have the Central African. So they all come together there in Kenya. So we would like to be able to build genetic profiles for all the black rhinos in Kenya, and we are currently talking to the Kenya Wildlife Services to see if we can get this done. And yes, as I said, we'll genotype individuals from Tanzania because we really want to try and maintain this Rovuma population. That's really important population if they still do have rhinos in that part. And then basically, from all of this, we have had the 7 subspecies being described by Colin Grooves based on the morphology and cranial skull and cranial morphology. Then we went to 4 subspecies, but using this historical data in conjunction with the extant data, we are kind of back to 9 sort of subspecies, of which 2 has gone extinct. This is the western subspecies that's gone extinct, longlipes, and then another subspecies here, Brucei, that's gone extinct, okay? The northeastern subspecies. But then we still have 1, 2, 3, 4, 5, 6, 7 subspecies that we think exist to this day, except for this Rovuma population that we are not exactly sure because no one has seen rhinos for a long time in this part of Africa. And all of this research has gone into this book. It's called Rhinos of the World. So they've kind of redefined sort of the number of subspecies and the genetic diversity that we have within black rhinos. At the moment, the African Rhino Specialist Group, they do not agree with this because they are feeling that a lot of the data that we've used, of course, to generate, this is based on historical samples, and those historical samples do not exist anymore. So the African Rhino Specialist Group, they are still standing by their 4 subspecies classification, and that is basically how they will use it in the future because they disregard sort of this historical diversity. But this is basically what we had in the past. We've had all of this diversity, and we've lost most of this diversity. So maybe then just one last thing to maybe try and make this a bit more practical. As I said earlier on, I am the co-chair of the African node of the Conservation Genetic Specialist Group. It's called ConGen Africa. We have a website. You can see here, our website is here at the bottom. And we are building sort of genetic management guidelines for, we've started with mammals, but we have just recently done our first a reptile, we have done a crocodile. So this is really answering the call of the conservation practitioners in South Africa specifically, because they really want guidelines, and especially for the translocation of larger mammals, because we move animals a lot across the African continent. You can buy, you can sell animals, so there's a lot of movement, and they want genetic management guidelines for the animals that they are moving. So here is the example of the black rhino. You can see here we had to adapt our map back to the 4 subspecies classification because this is what the IUCN accepts, the African Rhino Specialist Group. And then within our guidelines, we've built in these genetic indicators. This is the work that I have helped the JNCC produce for the UK. So we actually built this into our guidelines. And then we also include a bit of information similar to what they have in the Scottish scorecard. I don't know if you guys have heard of the Scottish scorecard, but that's sort of another metric, you know, building that into the conservation of your species and really talking about the genetic health of your population and what are the threats, you know, hybridization or genetic erosion, but including a little bit of that into these guidelines as well. So these guidelines are all publicly available. You can see here, we also have a book that you can download. We've currently done, I think we have done about 10 species that are uploaded. We are currently working on 3 new species. So we've just done white rhinos, and we've done hippopotamus, common hippopotamus. And then I have another MSc student that's currently developing 6 more guidelines for African carnivores. So it will include, we've done leopard already, but it will include lions, wild dog, cheetah, and then smaller carnivores such as serval and brown hyena and spotted hyena. So yeah, then it's just to thank, you know, the funders. So most of this work was actually funded by the Global Challenges Research Fund, but also the National Research Foundation in South Africa, International Rhino Foundation, and then also Cardiff University. And then I lastly just want to thank all my collaborators, people from the University of Pretoria back in Pretoria in South Africa, SANBI, which is the National Biodiversity Institute in South Africa, and they are advisors to the government in South Africa, even colleagues in Namibia, the Ministry of Environment and Tourism in Namibia, and colleagues even in Copenhagen. So yeah, thank you very much, and I'm happy to answer any questions.
Wonderful. Thank you so much, Isa-Rita, for such an engaging talk. We've got a few minutes for questions. I know you've got to run off at 2, but we'll see how far we get with them. I'm just going to quickly share it as well. So anyone who doesn't have access to the chat, if you're external, you should be able to submit questions to the Q&A using the QR code on the screen. So I'll jump right in. So our first question asks, what was the rationale for moving rhinos in the past, and would you advocate for any further translocations?
I think I am not a conservation, practitioner on the ground, and I'm definitely not the African Rhino Specialist Group that make most of those decisions. But I think it's a mix. I think sometimes there's some sort of, economic sort of motivation. It could also be political, and often they just don't know where to take these rhinos to, then they are sort of in a, it's a catch-22, we don't have space here, so maybe we should move them here. And of course they wanted to also rewild certain parts of Africa, where rhinos used to exist, but they had no more rhinos, so they just thought, oh, let's take some rhinos from South Africa. But now that we know this is the actual pattern, and these are very unique sort of clusters, genetic clusters, I would not advocate for this. And this is what we've been saying all along to the African Rhino Specialist Group, by using your 4-species sub-classification, you've actually already mixed things that you should have never mixed. For example, they only acknowledge sort of the Eastern African rhino, which is the Macaulay subspecies. But we know in that part you have the East African, you have the Central African, and you have the Maasai rhinos coming together. So by just regarding that diversity, I don't think it's very wise. But because the data that we show is mainly based on a lot of historical samples, they are arguing that we don't have this in the wild anymore, so therefore we can disregard it. And maybe there's some truth to that, but I think you have to take it with a pinch of salt, and I think maybe we should come halfway and they should come the other halfway, and we should meet each other sort of somewhere in the middle, I would say.
Thank you. So the next question asks, poaching of rhinos has reduced a lot over the last decade or so, according to your figure. Is this due to management efforts, or is it because there are just less of them now?
No, 100% management efforts. People have invested so much time and money into basically securing, their properties, the fences, they have patrol groups out, they have dog units, they have really invested a lot of money. So yeah, it's definitely because of the management and not just because there's less of them now.
Thank you. So the next question asks, how do you sample DNA from live rhinos, and do they need to be tranquilized?
Yeah, definitely. I mean, so when they do these translocation events, then of course they will always have a vet on site, because the vet needs to check the animal, the breathing of the animal. So generally they will take a little sample, then they can take a little bit of blood, they can take a bit of an ear notch, they can take a bit of rhino horn. In South Africa, a lot of properties, that's another thing that they are doing in South Africa, probably to combat the rhino poaching, is to actually dehorn the rhinos. So some of the smaller reserves in South Africa, routinely they dehorn their rhinos and they cut the horns. And when they do that, you can then pick up some of that rhino, horn shaving, because they just, they basically just use an electrical saw, and there's lots of, shavings, like bit of, it's like a hair basically, but just, compacted hair and nail. So it's also just creatine, and so they use those shavings and they put that in little tubes, and you can use that, for example, as a source of DNA. But generally, yeah, when you take a DNA sample from a rhino, you need to tranquilize it. But it's generally when they do other routine veterinary checks or translocations that you then have an opportunity to take a DNA sample. You don't just go out to take the DNA sample for the sake of taking the DNA sample.
That makes a lot of sense. Thank you. So we've got 2 questions left. So the first one asks, are there any ways in which genetic diversity can be increased naturally? For example, the introduction of individuals from north of the river boundary to the south.
You can, but you don't want to do that because you don't want to mix that, because historically they have never been in contact with each other, or there's been some isolation. I mean, the Zambezi-Chobe River would be able to dry up maybe from time to time, and so now and again an individual might move across that barrier, but historically, it has been very restricted gene flow. So I would not mix individuals from the north with from the south, unless there is absolutely nothing left in the world and there's nowhere else that we can go to find rhinos. Then you might want to do that sort of as a last resort.
Thank you. So our final question, are there any new or upcoming diseases that you're worried may threaten the rhino populations due to low genetic diversity?
No, not that I know of any diseases, unless you guys are aware of any disease, some mammal diseases that are popping up. I'm not aware of anything very specifically. And I don't think rhinos has ever really been hit hard by disease. I think their biggest enemy thus far has been us humans, poaching, and basically not just poaching more recently, and also maybe at lower rates, historically. But I think also another thing is like habitat, habitat fragmentation is a big problem. We were just talking about this the other day. This is now for white rhinos. Most of the rhinos that we have in South Africa are not in big sort of reserves anymore. It's like literally smaller little farms or nature reserves. So because there is nowhere else to take these individuals, they are all in this like small isolated little pockets. And that is then when you really have to start thinking about the management, because if you keep these pockets all isolated by themselves, they will become they will drift away because they are small populations, and they will become different from the rest of the population. So there you actually need very active management, and you need to move individuals. I don't know, you need to do the tests, but say once in a generation, or, every 2 generations, maybe one male, one female. You need to move them sort of to keep that gene pool homogenized across that sort of, as you can call it, a little meta population of smaller populations within the big meta population.
Wonderful, thank you. So that brings us to bang on time. So well done. So yeah, I'll end by saying just a final thank you again for such a brilliant talk. And thank you everyone for joining.
Yes, thank you very much for inviting me. I really enjoyed it. And yeah, if you have any questions, you can always email me with more questions. If you want to ask me something more specific, or if you want to actually have a chat about anything, I'm happy to do that too. Thank you so much.
Thank you. Bye, everyone.
Thank
you. Bye.
Bye.